In today's video at Medical Sciences, I want to discuss about drug discovery and drug development. This is covered under pharmacology, so we want to see how are drugs discovered and developed by the pharmaceutical companies. It is a complex process which requires more than 10 years. It ranges between 10 years to 15 years to develop a drug. So we want to look at step by step, how is a drug discovered and how is it developed. And as we begin, we remind you to subscribe, to like the video and share the comments and share to others. And we can also urge you to join in membership. This channel is attracting those who are interested to become members by subscribing a certain amount per month. Though there is that provision of 10K, those of 20,000, so that this work can continue to grow to serve the society. So we want to begin with different phases under drug discovery and development, and it is categorized or divided into three. So we are seeing it is divided into three, whereby we have drug discovery. And we are going to see that drug discovery will entail lead identification, lead identification, what does it mean? Here it is where we determine which chemical compound we are going to develop to be suitable to solve the problem or the disease in the society. So we first identify the disease or the target site we want to develop a drug for. So that is what we call lead identification. Then after identifying the candidate drug, we do what we call lead optimization. Then after drug discovery, the second phase shall be preclinical trials. And in preclinical trials, these ones are done on animals. These are trials done on animals, whereby first they are done on small animals, then later we perform them on large animals. And small animals include like mice, rats, and guinea pig. So these are the small animals we first do trials on. After passing, then we continue to large animals, and large animals includes cats and dogs. These are some of the examples where we do these preclinical trials. And what is the essence of doing preclinical trials? We shall see it will aid in many things, as we shall see. Then lastly, we shall look at clinical trials. Here, after you have passed the preclinical trials, we shall see you will make an application called IND. So this one they will give you an opportunity to continue to try this drug, the candidate drug in human beings, as we shall discuss it under clinical trials. So the first thing, let us begin with drug discovery. And we have said drug discovery, it entails lead identification and lead optimization, whereby under lead identification or finding, we are looking for that candidate drug which can target the desired, or which we can develop to achieve the desired goal. If we are interested to look for the drug or to develop a drug which inhibits renin-angiotensin-aldosterone system, then we have to find the candidate drug that is suitable. That is what we are calling lead identification, to come up with the suitable candidate drug. And it is determined in the following ways, whereby we have what we call random screening. We do what we call random screening. We do what we call compound. We do what we call compound oriented. There is what we call compound oriented. Then there is what we call target oriented and rational. Oriented. So these are the different methodologies or ways how we can identify the lead or the candidate drug. Whereby under random screening here we do several tests. Here several screenings or several lab tests are done to identify the chemical activity or the biological activity of the chemical. So here we are doing several compounds to determine the biological activity of the chemical. Here we want to identify the biological activity of the chemical which we want to be the lead. After finding the biological activity, another way of determining the lead is compound-oriented. And under compound-oriented, we shall see here, we use the existing drug or compound or chemical. So here we use the existing chemical or drug, and we could do what we call structural modification. Structural modification is done to come up with a suitable drug. So here we are using, for example, if we have penicillin, we can do structural modification to see if we can develop amoxicillin by just changing the structure appearance of the existing drug. That is what we call compound oriented. And after alternating or changing or modifying the structure of the existing drug, we can end up coming up with another lead candidate drug. Then another method we can use is target-oriented, and under target-oriented, we shall see here, you are developing a lead or a candidate drug which targets a certain molecular site. So here we are seeing maybe, excuse me, we see here you want to develop a certain drug that targets a molecular site, and this molecular site, for example, maybe you want to develop angiotensin converting inhibitors, or you want to develop, if you want to develop angiotensin converting enzyme inhibitors, then you try to target these ones, to see this drug will target what we call RAS system. So this is what we call target oriented. You know the molecular site you want to inhibit. So you test the chemical compound to see if it can inhibit that site. Then lastly is rational oriented, and under rational oriented, here we know the functioning of a particular system. Maybe we know there is what we call proton pump, which enables release of hydrochloric acid in the GIT or in the stomach. So if I know that if I want to target a certain pump, like maybe proton pump, or what you call potassium proton stroke potassium ATPase pump, and I know how it functions, here we develop a drug to inhibit this. A drug is developed to inhibit. So here it is, or maybe I know that COX-1 is an enzyme responsible for formation of prostaglandins and leukotrienes. So here I can come up with a drug that inhibits maybe cyclooxygenase type 1 or type 2 inhibitors. So this is what we call rational oriented. After understanding the working system or the mechanism, how the system works, you develop the drug to target or to inhibit that so that you can treat disease conditions in patients. So this is the summary where we can discuss about lead identification, or what we call lead finding. I told you that lead finding or identification here is coming up with understanding what drug you want to develop as a company and which solution you want to solve in the society, or which condition you would like to treat. After we have identified the lead, now the pharmaceutical companies go ahead to look at lead optimization. So under lead optimization, after coming with one or two promising candidate drugs, if you come up with one or two or more promising candidate drugs, you have, or lead, here we haven't even called it a drug, we have come up with one or two or more candidate lead, and this one, they are called lead compounds. These ones are called lead compounds. So this lead compound, after coming with it, it is further scrutinized, whereby several screening or several tests are going to be performed on this lead compound to come up with the suitable. So the major aim of lead optimization is to come up with the suitable candidate. Here it is, the major aim is to come up with suitable candidate drug. Suitable candidate drug. So that is the major aim of lead optimization, whereby we are seeing after coming up with one or three candidate drugs which are promising to be targeting a certain site or to treat a certain condition which we are calling a lead compound, we further optimize it by doing several tests to ascertain or to be sure which of them acts better or acts best to treat a certain condition. That is what we call lead optimization. After optimizing this lead and coming up with the suitable candidate drug, the second thing is now we have finished and we have optimized the lead, and now we are sure this certain drug candidate is the one that is going to work on or to solve our problem. So what they do, they go to the another phase which we know as the preclinical trials. That is the second phase. After the drug has passed through and you have come up with a suitable one, now we subject it to preclinical trials. And here under preclinical trials, we are going to do what we call in vitro studies. Here we are going to use lab animals. Here lab animals are used. We don't use a human being to experiment, but we use lab animals, and we are going to see we shall use different type of animals, whereby some we shall use we normally begin with small and we end up with large animals, whereby the small animals used include mice, we have rats, we have guinea pig are used. Then after the candidate drug has passed these tests, here we are looking at the pharmacokinetics, the general pharmacokinetics of the drug candidate. We are looking at the toxicity. We are looking at the therapeutic index of the drug under here. And even we are going to test the teratogenicity, the mutagenicity, and the carcinogenicity of that drug. After it has passed the small animals, then we subject it to large animals like dogs. We can use cats to do further studies. And as we have said that the major aim of preclinical trials is to come up with general pharmacokinetics, to come up with, to do toxicity studies, to do the therapeutic index of the drug, and under toxicity we are saying we can determine the teratogenicity to see if the candidate drug has effect on the fetus, or we can do carcinogenic studies to see if the candidate drug has ability to cause cancer. Then we can also do what we call mutagenic studies to see if the candidate drug is able to cause mutation in genes. So these are the major tests performed in preclinical, and these tests are performed on lab animals, and later the blood is collected and the tissues to screen for the effect of the drug on the vital organs, different toxicities formed. So this is done in preclinical. And after the candidate drug has passed all of this in preclinical trials, an application is made. When the drug is promising, if the candidate drug is promising and has passed, here you make what we call IND. that is investigational new drug. We call it investigation new drug. So you make this application to FNDA. It is done to FDA, that is USA Food and Drug Administration. So this is where you make an application for approval. So here you are looking for the authority or to be approved to go ahead to go into the clinical trials which are done on humans. After passing the tests done in animals, then you make an IND, that is investigation on new drug, to USA Food and Drug Authority for approval. When you're approved, that's when you proceed to clinical trials. And when you reach in the clinical trials, which we are seeing that these clinical trials are done on humans, these ones are not on animals but on humans, they are categorized into 5 phases currently. Formerly there used to be 4, but we have developed the 4th one, which is phase 0. So we are going to see under clinical trials we shall have phase 0, then we shall have phase 1, then we shall have phase 2, shall have phase 3, and phase 4. So these are the phases performed in clinical trials. But when you are reading different textbooks, many of them give phase 1 up to 4, but this one has been developed to prevent much money used or spent on phase 1 when the candidate drug cannot even pass phase 0. And phase 0 is known as microdosing. This microdosing is whereby the minimum drug dose is given to few people ranging between 1 to 10. We use normal human beings, 1 to 10 normal humans, normal human volunteers. These volunteers must be healthy. And here we exclude the elderly and the young children, whereby we use between 1 to 10 individuals who are volunteers, not sick, maybe exception of anti-cancer drugs which we use the sick people. Here we give them, we subject these volunteers on small doses like 1 out of 100, 1,000, 100 dose of the normal, or we use maximum of 100 microgram. Can use a maximum of 100 microgram of the normal dose. So here we are trying to give the smallest dosage to this normal human being so that we can study further the pharmacokinetics of the drug. We can look at the drug safety and sometimes the tolerability. But the difference is that we are using few normal human beings so that we save money or we prevent, so that when the candidate drug does not pass microdosing, here we cannot proceed to phase 1, which requires many people and requires much money. So after the candidate drug is promising, it has shown good pharmacokinetics in these individuals, has showed tolerability, now we proceed to phase 1, whereby under phase 1 it is also done on normal human beings, it is also done on normal human volunteers, and we normally recruit, the companies normally recruit between 10 to 100 people. So after recruiting these people, we give them this candidate drug, then we keep monitoring them to see the effect of the drug on this. And the major tests performed under here, we are looking for the safety, we are looking at the safety of the drug when given, whereby we shall determine its therapeutic dose and index. Then another thing we are looking for is tolerability, the tolerance of the drug, how human beings are able to tolerate, at which dose do they tolerate this drug without causing toxic effects on human individuals. And this one takes between the duration takes between 1 to 2 years. This is the duration, 1 to 2 years, to finish this study in normal human beings. And after we have determined that it is very safe, human beings can tolerate this chemical candidate or the candidate drug, and it is promising, we proceed to phase 2, whereby under phase 2, here we are going to use patients. It is done on patients, phase 2. And these patients, they range between 100 to 500 patients are recruited in this study. And these patients which are used in this study, this study takes between 2 to 3 years in these patients, whereby the major aim of this study is to look at, here we are looking at the efficacy of the drug. We are testing efficacy and potency of the drug in this phase. So here we want to look at which dose gives better results. So which dose can treat these patients continuously without causing adverse effects, and which dose produces toxicity. So here that is the major thing we are doing in phase 2, which takes 2 to 3 years, working whereby we are doing it between 100 to 500 individuals. So this is phase 2, what we are calling phase 2 of the clinical phase, or what we call clinical trial. So after determining the efficacy of the drug, we proceed to phase 3, and under phase 3 still we are doing it on patients. And these patients here we are doing on a large number of patients, between 500 to 1,000, or even greater than 1,000 are done. Patients. After identifying these patients, we use what we call double blinding. The process of this trial is done by double blinding, where the doctors and the patients, or what we call the researchers and the respondents, they are not aware of what is taking place. So it is a double blinding, and here we are doing it to confirm the efficacy of the drug. In phase 2, we are developing the efficacy, or we are looking for how candidate drug is efficacious. Then here we are confirming, and this takes between 3 to 5 years to complete. And it is done on multi-site. It is a multi-site study. It is done on different sites and ethnic groups, where you don't go to one place. You do one in certain place, you look for individuals of different genetic makeup and ethnicity, so that you can generalize and confirm the efficacious, or how the drug is efficacious in patients. So this is the importance of phase 3. So it is a multi-site trial done on different sites, which is different from phase 2. Phase 2 can be done on patients in one place to confirm the efficacy of the drug, to look at the efficacy. But when we want to confirm, we want to see, does this drug address the same, does it solve the same solution to people of different genetic makeups? Does it solve the problem of the Indian the same as maybe Africans? So we do on different sites. It is a multi-site study, and that's why it takes long, so that we confirm the efficacy of the drug. Don't forget that. And when the drug is promising at this level, that's when you make another second application, and the application made here is known as, it is made under phase 3. Here, when the candidate drug passes the efficacy and is confirmed, you make a second application called NDA. That is New Drug Administration, whereby this New Drug Administration is also taken to Food and Drug Authority or Administration, that is USA, so that they can give you a go-ahead to start marketing this drug. So you make an NDA application, New Drug Application, so that you can be approved by the FDA. After being approved, that's when your drug is started to be marketed. So this approval is to enable the drug to start to be marketed, to be on the market. When you are approved by the FDA, that after forming New Drug Application to FDA, here when it has passed and you are approved, you begin now marketing the drug you have developed. And this one also approval takes between 1 to 2 years. So we are seeing this drug development section is very hectic. Then lastly, when you start marketing and the drug is on the market, people are already consuming it, then this one will lead us to phase 4, whereby this is known as post-marketing. This is known as post-marketing study. And this one does not need any ethical consideration by ISH of good clinical practice. This one does not need, it doesn't need ethical consideration by the good clinical lab practice and ICH. So if they ask you the phase which does not need clinical consideration, that is phase 4, which is post-marketing. And here we are looking for the rare adverse effects of the drug. We are looking for the rare adverse effects of the drug. That is what we are looking for. And it is not time bound for it, the continuous, as long as the drug is on the market. This is a continuous process where we keep monitoring. The pharmaceutical companies keep monitoring this drug to see if there are rare conditions or rare adverse effects this drug is bringing on the people, on the patients. So this is what we call post-marketing phase. And in case they find that this drug is causing some rare adverse reactions, it can be held or it can be called back off from market. As we saw, like aspirin, we saw a drug like chloroquine, which used to bring much adverse effect, which was later stopped. That was during post-marketing phase. When we find this candidate drug is causing some rare adverse effect or toxicity on human being, which were unnoticed during these first 4 phases, then it can be called on market. That is the instance of post. So this one, it deals with pharmacovigilance. It deals with pharmacovigilance. Pharmacovigilance is whereby, as we are taking these drugs, we keep monitoring to make sure we are taking the right dosage, the right indications and the contraindications they are put in place, so that this drug we can see if we can identify some adverse reactions so that this drug can be stopped. And we said this is not time bound and does not need any ethical considerations. So this is what we can discuss under drug discovery and development. And in case you asked some multiple choice questions, maybe they ask you that in which phase do IND application made, that is before clinical phase or before phase 0 and phase 1. That is after preclinical trials. And in case you are asked that in which phase of clinical trial do we find or do we study drug safety and tolerability, that is in phase 1. And in which one involves microdosing, that is in phase 0. Then that which of maybe phase use normal human volunteers, that is phase 0 and phase 1. Then phase 3, phase 2, 3, and 4, for them they use patients purely. And if they ask you that which of the following phase do we see determination of efficacy, that is phase 2, then confirmation of therapeutic use or efficacy, that is in phase 3. And if they ask you in multiple choice questions that which of the following phase of clinical trial do we see application of NDA, that is a new drug application, to FDA, that is at the end of phase 3 of the clinical trial. And the post-marketing or pharmacovigilance is performed under phase 4. So these are some of the multiple choice questions that can be asked when you are in an exam. So thank you so much for learning with us at Medical Sciences, and may the God bless you.